myself and turn off the camera. I'll be here to try to help if issues come up. Thank you.
Thanks, Meg. So my name is Abdinur Abbas. I'm a professor in the Department of Bioproduct and Biosystem Engineering. And my researcher today, actually, Zarori, will be presenting some of our work related to invasive species, technologies to fight invasive species. We started this project a couple of years ago, and we had the continuous support of the Center for Aquatic Invasive Species. And during the pandemic, we actually used our work on this project to develop a kit to extract RNA from, for coronavirus for COVID-19 diagnostics. And we were successful in developing a kit that performed better than commercial product. And right in the last month, we came back to the original project of invasive species. And the main objective is to develop technologies to try to get eDNA from water, so we can use it to evaluate the presence and distribution of invasive species. Actually, is my researcher who works on this project. He started here a year ago. He has already a background in ecosystems engineering, and he has been developing this technology, and he developed also the technology for coronavirus. So I will let him introduce his work and talk about some of the results we had in the last year, and then you will have a chance to ask a few questions. Go ahead, Aki.
Thank you for this nice presentation. So I will try to share my screen, but I can't share my screen. Don't know why. Yes, so we can.
Can you share it now?
Share out.
Okay, good. We
Yes.
can see.
Let's see. So, hello everybody. Today I will present my work about novel technology of eDNA collection and concentration. 137 billion per year is the cost or impact of invasive species in USA. So it's a huge amount of money caused by damage caused by invasive species. So first I will start by defining what is environmental DNA, and I will describe our technology for DNA capture, and then I will present results and discussion, and I will finish with the conclusion and outlook. What is environmental DNA? All living organisms release DNA into its environment, and there is we can find DNA in tissue or free DNA. I mean extracellular DNA. Imagine we go to the lake and would like to know if there is invasive species or to know what live in this lake. So we have a choice between two methods: the biomolecular method based in eDNA, or traditional method based in observation and determination by visual observation. So there is a difference between the method because eDNA has advantage. We can target many species at once, and it's allowed us to detect invasive species or rare species early than the traditional method. So, for example, to study biodiversity or bioconservation or invasive species control, we need to get DNA from the lake. For this, we need to capture it and to detect it with qPCR or genome sequencing. These techniques, I mean qPCR and genome sequencing, are powerful and sensitive, but it depends on what we captured. So there is limitation in filtration. For example, we cannot capture free eDNA, and there is inhibitors and clogging, and there is also a problem in DNA conservation. So there is a solution. We propose this technological solution based on sorbent for eDNA. So here we have a filter, and we functionalized with positive polymer, and then we can capture eDNA, which is negatively charged by electrostatic bond to our positively charged filter. As you can see here in SEM images, we have in the bottom, in the top, cellulose filter, and in the bottom, functionalized one. And you can see the difference between the two. We have the polymeric nanoparticles developed by functionalization. And this gives affinity to our filter to capture DNA. To test this, we tested our sorbent and compared it to nitrate cellulose filter and cellulose filter. These three filters have almost the same pore size, between 5 and 6 micrometer, and we tested by free DNA to study loading capacity. The loading capacity is how much the filter can capture DNA. So as you can see in this figure, we have for the nitrate cellulose and cellulose filter, the amount of captured DNA is insignificant. For our filter, we can capture up to 381 microgram of DNA per gram of sorbent. For assuming that the DNA concentration is around 10 nanogram per milliliter, one filter can filtrate and recover the DNA from 38 liters of water. And considering like the water quality is good. Here, I will show how our filter can work. So in the field, we can just put the filter into the holder filter, and we pass the lake water through it. And you can see here, the DNA will be like absorbed into the filter. And in the lab, we can elute the DNA in 1 milliliter. So we pass like from 1 liter, if we pass 1 liter of water into the filter, to 1 milliliter. So we concentrated DNA in 1 milliliter. And then by purification, we can reconcentrate DNA in 10 microliter, and then the DNA will be ready to be analyzed by qPCR. Here we studied, we used herpesvirus, and we used very low concentration of DNA, and we tested it as previous filters, so nanosorbent and cellulose filter and nitrate cellulose filter. As you can see, it's the same results for cellulose filter and nitrate cellulose. There is a significant capture, and for nanosorbent, we capture up to 100% of DNA, and recovering from the filter is around 30%. So we successfully concentrated DNA from 0.7 copy per microliter to 84 copy per microliter. So we concentrated DNA more than 10 times. As a conclusion, we developed a new eDNA kit and which is able to capture 100% of free DNA and to concentrate free DNA up to 300 times, and it conserves eDNA in the filter in solid state. As impact by this performance, we can capture more DNA, and we have access to lacking information by capturing free DNA, and this give us more accurate information about the species living in the lake, and this can reduce also this because our filter is performant and can reduce the sampling effort, and also it can reduce the cost over 5 times than the available kits. We optimized buffers for coronavirus, and we are still working how to recover eDNA tissue. So we are working on it, and we need further validation with CARP, viral DNA collection with Professor Phelps, and soon third-party testing will be done, and we can have like validation, field validation of our kit. So I thank our collaborators and our sponsors, Minnesota Aquatic Invasive Species Research Center, and environmental and natural fund, trust funds, and University of Minnesota Drive and Discover. And thank you for your attention. And if you have a question, I'm ready to answer them. Thank you.
Thank you, actually. We don't have a lot of time, so we're going to just go straight to the question. And I think the way it works is that you write down your questions in the Q&A you have down there, Zoom, and I will ask the question, and I'll see, actually we'll have time to answer them. In the meantime, I have one question for Akli.
Yes.
What is the major advantage that your filter, major problem that your filter solves compared to the commercially available ones? If you can name one major advantage.
So the advantage of our filter now is it's like we opened a new path or access to exploit free eDNA. So the all kits available now, they haven't access to exploit or to capture free eDNA. And the second, I think it's also the cost, because we master this technology and we can fabricate our filters and our buffers. So the cost will be like divided into 5, something like this.
Okay, thank you. I have a question. Does this work for any species in the same way?
Yes, it's like it works with the same like principle, but for example, for vegetal and animal cells, or for DNA, it's the same. For free DNA, it's the same. But what we are developing now for like buffers for tissues. So for tissues, vegetal and animal are different. So buffers can be different, but like globally, the technology it's the same.
Okay. Okay. So the technology works for any species.
Yes.
In case you change from animal to something.
Vegetal, we have to do some.
Then you just change the buffer for that.
Yes,
okay.
Yes.
Another question is, can citizens access test filter and use a third party to test those samples?
Sure, yes, because our filter is easy to use, and it's easier than other kits available now. So yes, citizens can have access to use our kit and to explore the result for the third parts. Yeah.
Okay, thank you. And I would just add to that that once those kits are ready and they will have their spec sheets and with their on the steps on how to use it, we will be sending them out to anyone who's interested to use them. And of course, you just collect your sample and then you send it to another lab to do qPCR if you don't have the ability to do it in your lab. But they will be available for free to test by any lab who is interested on trying them. I have another question here. Is sample processing quicker? And where is the cost saving over other methods?
So yes, for this we have to do some experiment, because our filter is efficient, so it can capture even free eDNA. So for example, as we can see from the experiment we did, for normal filters, it needs perhaps more time or more water to filter in order to have a small amount of DNA. But for ours, because it's very efficient, so it can just small amount of volume of water, and it capture a lot of DNA. So the sampling effort it consists in this.
Okay, so the cost saving comes from the fact that we won't have to use more filters to filter the same amount of water. But the other part of the question is the sample processing faster? Like when you do the filtration, yes. Is it faster compared to QIAGEN kits? Or?
Yes, because we have like we have a part we can do in the field. We filter water and then we can elute the DNA directly from the holder filter without taking off the filter from the holder. So in the lab, we can just elute it with a specific buffer. Yeah, so it's, for example, until this time, for QIAGEN, they have to recover that, the filter, and in the lab, you have to cut it into small pieces, part, and then they do like incubation for 3 hours, something like this, in order to have like to get DNA from the filter. So with our technology, you can save time, many hours in the lab and even in the field.
Okay, I have one question here. Where does your data get shared and who owns the data? I think that's a general question. This research is funded by the Center for Aquatic Invasive Species, but any research done at the university, the work is owned by the university, of course. And if there's any invention, then it's partly co-owned by the inventors and the university. And the data will be shared through publication and will be available to the scientific community and to the public. Let's see if we have another technical question. I have been doing plankton net, concentrating samples for visual review by third-party lab for zebra villigers. Can I use eDNA for that?
Yes, sure. For each like invasive species detection, you can use like eDNA technique. Yeah, we can use that. Sure.
Yeah, I think not only aquatic invasive species, but basically any species can be detected if we analyze the eDNA released. I think there is still work to be done by the scientific community to try to correlate the abundance of species with the concentration of DNA we detect. But I think it's a very powerful technique that can be applied to any species.
Yeah, exactly.
When will the filter test kit be available?
Yes, so this is need like it's need validation. So the first validation we can do it with Professor Phelps to study like the relationship between herpesvirus and carbs, and then once we did it with this, we will test it with the third party, and then it will be available like perhaps by the next summer for commercial.
So just to answer clearly this question, the plan was to get it to our partners for third-party testing this summer. That was the plan before the pandemic, and we did a lot of work on that. And when the pandemic started, so basically over the last 5 months, we applied all the work we had to detect COVID-19 coronavirus. So we developed the kit that's actually ready to go to the market. That actually went to some collaborators already, and that's ready to go to the market today, focused on coronavirus detection, extracting RNA from coronavirus, recovering the RNA, and then doing RT-PCR. That one is ready to go. So now that the pandemic is slowing down, hopefully, we are going back to the eDNA, and we hope that within probably 3 months, we can start sending kits to our partners, a couple of months, sending kits to our partners for third-party testing, and hopefully early next year, it can be ready for go to the market.
And we have a fund like independent from NSF for the
so basically the pandemic slowed down this project around 5 months. That's why we don't have it ready this summer. Have another question here. Was the Nanosorbent developed specifically for this project, or did it exist previously? So we had the technology before this project, and then we tried to apply it to, so the patent for this technology existed before, but it wasn't applied to invasive species. So we started applying it to invasive species, and then the pandemic started, so we started applying it to the coronavirus. We finished that project with NSF funding. So we had NSF funding, COVID-19 related funding from NSF, and we finished that project. And now we're going back to applying the same technology to invasive species. And hopefully within a couple of months, we can come up with viable tests that our partners can try. I'm going to give it a few seconds if there's any more questions, and then have you run into roadblocks pertaining to the ethics of this work? I think the question needs a little bit more clarification on what kind of ethical problem this may cause. We'd like to give them a clear answer, but it would be better to understand what kind of ethical issues we're talking about here.
Perhaps there's some confusion about this, maybe getting confused with other genetic control methods, and maybe I don't know if you want to elaborate on how this is really just sort of a surveillance.
So I had a comment that's a little bit different. So it says collecting DNA from water being on a reservation land. Personally, I really don't know how to answer that question because I never dealt with that kind of situation. But I guess if anything is done in a reservation land, it should be done within the regulation of the state on that. So I cannot really answer this question. We haven't been faced with this kind of situation so far.
I could just chime in and say that any MESRG researcher that was collecting samples on tribal lands or within any kind of jurisdictional boundary that a tribe manages, we would only collect samples with the full permission and support of the organizing entity and the folks that are in charge of those decisions. So I hope that allays any concerns, but at MASERC and the U, we're very aware of the potential conflicts and would only move forward with permission from the tribal authorities.
Yeah, thanks, Meg. Okay, I think we are done here. And if you have any more questions, please feel free to send them to me or to Akli. You have my emails in the presentation, and you can also get my emails from Meg or from the center. We'll be happy to answer them or have a conversation with you about more details on this project.
Oh, Abner, can I just say, jump in? We did get one more question, and
yes.
It's a really good one. So I think it'd be good to address.
So I see a question. Is it, can eDNA define the quantity of an aquatic invasive species or just the existence? Is that the one you're talking about, Meg?
Yep, that's the one.
Okay. Can eDNA define the quantity of an aquatic invasive species or just the existence of those species?
It's both, because many articles, paper made links between the quantity of eDNA recovered and the abundance of the population. So it can do both, so detection and also quantification of the species.
Yeah, and I would like just to add to that, the fact that our sorbent, our filters, we call it sorbent because it's not really a filter, is that if you capture all DNA, free and tissue DNA, you can also not only have the presence of the species or quantification, but you can also have a chronological presence of the species, because free DNA, which means it's a species that was there earlier and the DNA is already in free state. If a species is there later, then you will have more tissues and more cell. So if you are able to analyze free DNA alone and tissue DNA alone, you can have more information on the timing on the presence of those species.
Exactly, yes.
Oh, and just see, we did get another question from Mike Sorensen. This is a good, Mike has done some eDNA surveillance work with the Minneapolis Parks Department. So he's got some good expertise here. It's in the Q&A, Abdinor.
Okay, let me, I see it. Okay, did you find that your filter was clogging after a similar volume of filtered water as other tested 5 to 6 micrometer filters? I have been collecting eDNA samples from fairly productive lakes in late summer and find that the filters clog after I have only filtered a few hundred milliliters of water. I'm wondering if your new filter will help with this issue.
Yes, for this, I think the clogging would be, because the clogging is not from getting from eDNA or from just from tissue with targeting. It's also coming from undesirable or unwanted particulate in the or from suspended material in the water. So it will be the same, but the good thing with our filter is with the same volume. For example, if with the clogging occur at 500, for example, 500 milliliters, with this same volume, we can capture more DNA. But the clogging, I think we will face the same problem because the clogging is related to the pore size. But we can, for example, test to functionalize larger pore size and, for example, 11 or 20 micrometer. And the plugin, we can enhance capturing because our filter has affinity with the DNA. So we can enhance like the volume we would like to filter with
so
this.
Just to follow up on that answer, that's the whole difference between a filter and a sorbent. A filter relies on pore size to capture something. So that's why people need to use natural cellulose filters that are small size, 5 micrometers or 2 micrometers, to be able to capture DNA. If you put 10, the cells are going to go through them, or DNA is going to go through them. The difference with solvent, a solvent doesn't rely on pore size. Solvent relies on affinity with eDNA, which means we can play on pore size and make it even larger in a way to have faster flow rate, in a way to have less clogging, because the larger the pores, the less problems you have with clogging. So we have the ability to do that and have solve the clogging problem, or at least to reduce it significantly, while keeping the same ability to capture DNA. If we don't have any more question, do you see any, Meg?
Nope, I think we got them all.
Okay.
Thanks.
Yeah, I think we had very interesting questions. Some of them already addressed the problem scientifically and we solved them. Some of them we're still working on it, but hopefully what we're trying to do is to get the scientific community working on invasive species, but also our stakeholders or even citizens, get them a tool that they can easily use, that's very cheap, and that's very efficient on capturing eDNA, because I believe that eDNA is the ultimate tool to try to monitor the environment, whether it's aquatic invasive species or large species. But the problem we have been facing for the last years is that we have a great detection technique. qPCR, genome sequencing is a huge revolution. The problem is you can only detect what you capture. And the problem we had for the last years is we didn't use any new technologies to capture DNA. We have been using filter paper, like normal filter paper that you can find anywhere. There wasn't any technology development in that field. And that's the problem we're trying to solve now by making a technology specific to capture eDNA. And if we have all the eDNA in the water when you do your sampling, then your data, your information with qPCR will be much more reliable, and it would be much more useful for decision making. If you don't have any comment, Meg, I think
that's everybody. Yeah. Thank you, attendees, and thank you, presenters. That was great.
Thank you very much. Thank you, Akli.
Bye, everybody. See you at the next one.
Thank you.
Have a good evening. Bye.
Bye.